welcome to the video for 6.3. So in this video, we're going to talk about microbial nutrition. So things that bacteria need to eat. Okay, so we're going to start with a review question. Surprise. Okay, so we have a culture of bacteria that is growing exponentially under ideal conditions. Its generation time is 20 minutes. If the culture starts with 2 bacteria, how many bacteria will you find after 1 hour of growth? Is the answer A, 2, B, 4, C, 8, D, 16, or E, 32? So pause the video, take a second, do some math, and see if you can figure out what the answer is. Okay, so the answer for this is D. Now, we could do it the regular way where we just double it. Let me turn the little pen on here. Okay, so we start with 2, we double once. Okay, so 1 generation time. We go to 4. Okay. Oh, we probably should talk about how many generation times we have. So if our generation time is 20 minutes and we go for 1 hour, that means we have 3 generation times. Okay, so we go from 4 to 8 in the second generation. Then in the third generation, we are going to go to 16. Okay, so we can do the math that way. Let's erase that. Okay, or we can just use our equation, right? So we have our n0, which is 2. So we're starting with 2 times 2 raised to the number of generations, which is 3. Okay, so basically we have 2 times 2 times 2 times 2. Okay, so 2 times 2 is 4, times 2 is 8, times 2, this is 16. So the answer is D. Okay, definitely know how to do this for the next exam. Okay, so let's start into section 6.3. So our section objectives, we're talking about microbial nutrition. You need to be able to compare heterotrophy, autotrophy, phototrophy, and chemotrophy. Lots of trophies. Okay, and you need to be able to describe the importance of the nitrogen and carbon cycles and the roles of microbes in their maintenance. Okay, but first, we need to talk about microbial growth. Okay, so let's think about a scenario. Okay, so we have ideal conditions, no limits on bacterial growth. What do we see? So one E. coli bacteria cell can divide into 2 every 20 to 30 minutes. Now, if we use the 30 minutes, so the longer generation time, well, how many times can we divide in an hour? We can go through 2 cell divisions in that hour. Now, if we think about a whole day is 24 hours, so that means we can go through 48 generations of E. coli in a day. Okay, so that 1 E. coli cell, so I did the math, so that 1 E. coli cell in 24 hours can become 218 trillion cells in 24 hours. But the thing is, about 100 to 100 trillion cells weighs about approximately 1 gram. So after 24 hours, we get about 2 grams of bacterial cells. Okay, but that's 218 trillion is what we have going into the next day. So if we go for another 28 hours and have it go for 48, well, that 218 trillion after another 24 hours is going to be a very, very large number. Okay, so we go from 2 grams of cells after day 1, and by the end of day 2, we have 10 to the 14 grams. Okay, this is like roughly the weight of 4 million dump trucks. That's a lot of E. coli, right? But obviously this doesn't happen, right? We're not overrun with E. coli. That is because microbial growth has limits. Okay, we can only get up to these numbers. We can only get up to 4 million dump trucks, which is a lot. Okay, if if we have no limits. But in nature and in real life, we do have limits. Okay, we have things that are stopping microbial growth, so microbes aren't taking over the world. Because could you imagine? Could you imagine that many bacteria that they're the weight of 4 million dump trucks after 2 days? That's a lot. Okay, so we talk about in nutrients and environmental niches. So a niche is anywhere a living organism can live. Okay, so humans have environmental niches, right? Because we have more people that are living in Idaho and being successful and surviving than, say, people that live in the Sahara Desert. Now, there's people that live in the Sahara Desert, just not a lot. Okay, it's not a really good environmental niche. So bacteria also has environmental niches that we need to talk about. Now, if you took biology, especially if you took biology 201, you know what I'm going to say next. What does essential mean in biology? Essential means that something can't make something, but they can get it from their environment or get it in their diet. Okay, so microbes have essential nutrients that they can't make themselves, but they can gather from their immediate environment in order to grow and divide. Now, all living things need carbon, they need nitrogen, they need phosphorus, they need hydrogen, they need oxygen, they need sulfur. Okay, these are our essential elements. We have to get these in our diet. We have to get these from the environment. Okay, we also need things like magnesium 2 plus ions. We need iron 2 plus ions, especially as humans, because we depend on that 2 plus ion in order for our red blood cells to work and to gather oxygen. Okay, we need potassium, so K plus. We also need trace elements like copper and cobalt and zinc in order to function correctly. Okay, we can't make these elements, but we can get them from other things. Okay, that's why they're essential. So bacteria, they need these things in order to grow and divide. Okay, so in the previous, not last section, but in 6.1, we talked about different types of culture. We talked about synthetic versus complex culture. Okay, so when we're talking about nutrients, our synthetic media is chemically defined, meaning we only put in what the bacteria need to grow. Okay, so for E. coli, we'll need to make sure we have everything from the previous slide, plus glucose to act as a carbon and energy source. Okay, so that's what we see here in this table with the M9 media. Okay, this is what we know as a defined. We know everything that goes into it and how much. Okay, so in this media, we have 2 grams of glucose. We have 6 grams of sodium biphosphate. We have 3 grams of potassium biphosphate. We have 1 gram of ammonium chloride. We have 0.5 grams of sodium chloride. We have 2 millimolar of magnesium chloride, and we have 0.1 millimolar of calcium chloride. Okay, so, and we know that it's a pH of 7. We know everything that went into this. That's why it's defined. Okay, now we also have complex media, which we don't know everything that's in it. Okay, it works and it works great, but we just don't know why it works. Okay, so this is going to be when we use things like beef extract or yeast extract. So this first one in this table is lysogeny broth. We call this most often than not LB broth. Okay, it's also referred to as Luria bertani broth. Okay, so this is complex. So this is going to have about 10 grams of bactotryptone. Okay, so tryptones are peptides, so they're pieces of a protein that were made when an enzyme called trypsin broke apart casein, which is a protein. Okay, so we don't really know what are in these peptides. We just know that we have them. Okay, we also have bacto yeast extract, where we go and grind up yeast and add it to the media. And we have about 10 grams of sodium chloride and a pH of 7. So this would be a complex media because we really don't know what's in the yeast extract. We don't know what exactly is in the tryptone and the bactotryptones. We just know that it works. Okay, one thing I would be able to do is be able to look at a list of ingredients and figure out the media is chemically defined, okay, or if it's a complex media where we don't know everything that's in it. Okay, so this brings us to nutrients and environmental niches. Okay, we saw in the case study at the first of the chapter that our Haemophilus influenza needed specific growth factors in a plate in order to grow, or they grew on the chocolate agar but not on the blood agar. So why is that? That's because Haemophilus influenza needs things that they have to be able to gather in order to grow and divide. So things like pathogens, they're going to be stealing these growth factors from their host in order to grow and reproduce. So like the Shigella toxin that's going to cause bloody diarrhea uses NAD, so nicotinamide. Okay, we'll talk more about NAD in a later chapter and exactly what it is. So this next one is Haemophilus influenza. Okay, they need NAD like Shigella, so that needs to be a growth factor, but it also needs this hemin. So hemin is what you get when you lyse or break apart red blood cells. Okay, so this is why Haemophilus influenza grew on the chocolate agar, is because we had already lysed the cells and we had released the hemin into the agar, so they were able to use it and they were able to grow. Versus with the blood agar, the red blood cells were whole and intact, and Haemophilus influenza wasn't able to lyse the red blood cells to get to the hemin, so it couldn't grow. Now, we have different bacteria that could have grown on the blood agar because they are able to break apart the red blood cells, but Haemophilus influenza can't. Okay, then Legionella pneumophila, we've talked about this, Legionnaire's disease. Okay, this is the one that's in, say, like hot tubs. Okay, it needs cysteine in order to grow. Okay, mycobacteriums need some interesting things. We haven't grown leprae, so Mycobacterium leprae, the one that causes leprosy. We haven't been able to culture it yet, but we've been able to culture Mycobacterium tuberculosis. Okay, it's going to need nicotinic acid from NAD, and alanine in order to grow. Okay, so because these pathogens need specific growth factors, we can utilize this list in order to identify these microbes in the clinical setting. So we're going to have different petri dishes that are going to have different growth factors, and we're going to see what grows on them and what doesn't grow. That's going to help us eliminate what the pathogen could be. Okay, so like with Haemophilus influenza, well, it didn't grow on sheep blood agar, but it grew on the chocolate agar. So we know that it needs hemin, so it's going to what's the word? It's going to take our list and break it down into ones that can and can't. So our list is going to get smaller and smaller, so it's going to be easier to identify what pathogens causing that disease. Now, there are some bacteria we have no idea what they need to grow. We can't grow them. Okay, whether we don't know their nutritional needs, or the fact that we can't grow them in culture because they're going to need something like a cell in order to grow, because we have a lot of pathogens that have to have a host in order to grow. So one of these are going to be rickettsia. So rickettsia is a type of bacteria that we know grows, but we don't know what they need. We don't know their nutritional needs. Okay, and we can't just grow them on a petri dish. So rickettsia is actually going to require a human cell line, and we're going to grow them in cells in the petri dish in order to study them. This is really hard to do. I like hard things, but I run away from trying to grow things in cell lines. That's not for Miss Harker. Okay, so being able to study rickettsia is actually really hard because we have to have them in the cell in order to study them. Okay, so let's talk about the 3 things that all microbes need in order to grow and divide. Okay, so we're all carbon-based life forms. Okay, we all need organic compounds. Remember, organic compounds are just things with carbon in them. Okay, we have to be able to get carbon compounds in order to sustain life. Okay, so both simple and complex carbon compounds are used as food. We use them to store energy, like in lipids. Okay, and those hydrocarbons. And we also use carbon as a building material to make biomass. So biomass is just how well we can produce everything inside of the cell. Okay, so autotrophs and heterotrophs are two two types of organisms that obtain their carbon in different ways. So whenever you see troph or trophy, this is not like when you go and win a competition and get a nice trophy to put on your dresser. Trophy in these terms means feeding or eat. Okay, so auto means self, so self feeding. Hetero means other, so other feeding. So autotrophs make their own carbon compounds. Okay, they always usually start with CO2, so that's a good indicator of an autotroph. We have other types of autotrophs too, but we'll talk about those in a few slides. Okay, so an example of an autotroph would be photosynthetic cyanobacteria. So cyanobacteria are how we think that we got a lot of the oxygen in our atmosphere initially. Okay, so they are going to take carbon dioxide and use photosynthesis and light from the sun, and they're going to make glucose from that, and then they're going to use the glucose they made to power their cells. Now, heterotrophs means it feeds from something other, so we have to get it from outside of ourselves. So heterotrophs, this is like you and me and E. coli, okay, they are going to obtain carbon compounds from other organisms. So this can be either by eating autotrophs or eating other heterotrophs. Okay, heterotrophs often release carbon dioxide. Okay, so one of the things that we need to know about the last few slides is we're going to be talking about a lot of cycles, like the carbon cycle and the nitrogen cycle. So we need to kind of understand what this means. Okay, so this is a simple picture, too simple really, but it helps us understand. Okay, we all know, well, hopefully we all know by the time we get to biology or microbiology that life on Earth gets all of its energy from the sun. Okay, so the sun provides energy in the light rays. Plants are going to capture that light and they're going to use photosynthesis to make oxygen gas and sugar. Okay, so they make their own food. And then heterotrophs are going to come and eat the autotrophs and other heterotrophs. Okay, so if we look at this picture, we would think that once it hits heterotrophs, we're done. This isn't really a cycle. Okay, it goes from sun to autotroph to heterotroph. But this is, as I said, a little bit too simple of a picture. So let's look at what really happens. We need to talk about the global carbon cycle. Okay, so we have, I know this is a pretty terrible image, but it was given to us by Dr. Burroughs, so we're going to use it. So at the top here, we have autotrophs, and here we have heterotrophs. So autotrophs are things like plants, algae, many bacteria. Okay, so a lot of them will go through photosynthesis, but we have some others that get nutrients other ways. Okay, we have heterotrophs, that are things like animals, like us, fungi, and some bacteria are heterotrophs. Okay, so let's start with the autotrophs. They're going to get CO2 from the atmosphere, and they're going to harness the energy from the light or from other chemical compounds, and they're going to use that to create sugar. Okay, so we're going to make our C6H12O6. Okay, during this process, we make oxygen gas and sugars that contain carbon. Well, heterotrophs use oxygen gas. Well, a lot of them use oxygen gas, some don't. Okay, but they all use these carbon compounds. They use these sugars. So they're going to ingest them, and they're going to break those sugars down into CO2 and water. And both of the CO2 and water are going to go into the cycle and be used by autotrophs again. And it's just going to keep going round and round and round, feeding the cycle. Okay, so we get all the energy from the sun, but autotrophs are what harness it and use that to take CO2 from the atmosphere, because we can't directly use CO2 to make glucose ourselves. So it's going to take this inorganic, that's what we call CO2, it's inorganic, even though it has carbon, and we change it into an organic compound. So a compound that contains carbon that we can use. Okay, so this is the global carbon cycle. Now, the carbon cycle is pretty cool because it needs both autotrophs and heterotrophs in order to function, and sunlight. Okay, so let's start down here with autotrophs. This is my preferred cycle, but we're also going to look at some other things too, because we're going to have light absorption. So we're going to have the autotrophs that go through phototrophy, aka they go through photosynthesis. So they use light energy to capture to make glucose. We also have something called lithotrophy, where we're going to get our carbon from other places and our energy. Okay, but we're going to get our carbon source. Okay, we're going to take CO2 and water, and we're going to convert it to organic carbon compounds like glucose, and that glucose can go over here and go into the cellular respiration cycle, and heterotrophs can use it to make energy. We can make fermentation products. Okay, we can make ATP. We'll talk a lot more about ATP in another chapter. Okay, we can also, heterotrophs can use things like lipids and peptides and lignin in order to make energy and to use these for carbon. But the result is, either way you come into in with heterotrophs, we're going to make that carbon dioxide and that water that's going to go back and be used by autotrophs in order to make glucose again. Okay, so I kind of understand the carbon cycle here. Okay, so the first thing we have to do is for an organism to replicate and to divide and grow, we need to get carbon. Okay, but the next thing we need to do is we need to capture energy. Okay, so energy is essential for all forms of life. Life can't function without energy. So for bacteria, they can get energy from light or chemical compounds to power living things. Now, where you get your energy is going to tell you what another type of trophy you're going to use. Okay, so phototrophy. So phototrophs use light as an energy source. Okay, autotrophs are phototrophs. Oh, okay, so wait. Let me rephrase that. Phototrophs are autotrophs, so they can make their own energy. Well, then we have chemotrophs that are going to use the potential energy stored in the bonds of chemical compounds as their energy source. Okay, we have two types of chemotrophs. We have the organotrophs. This is what we are. We're going to use organic chemical compounds, so things with carbon in them, aka sugars, and we're going to break them down to make energy and oxygen gas. Okay, lithotrophs, on the other hand, are going to use inorganic chemical compounds. So things like hydrogen sulfide. They're not going to use sugars. Okay, actually, lithotroph is like one of my favorite words ever, because it literally means eats rocks. Litho means rocks. Troph means feeding, so it literally means to eat rocks. Okay, so phototrophs are autotrophs. So you would assume that chemotrophs would be heterotrophs, but that's not how it breaks down. So lithotrophs are autotrophs like phototrophs. Okay, whereas organotrophs, these are going to be heterotrophs. Okay, so I would know these terms. Okay, so we can take each of these terms and we can use them to describe how organisms eat, how they feed. Okay, so auto and hetero is going to describe where we get our carbon source for biomass. Okay, auto is going to use CO2, and heterotrophs are going to use a carbon source found outside of them. Okay, so when we're trying to identify auto versus heterotrophs, we're going to look for whether it fixes CO2, so whether it uses CO2 to make its own food. Okay, the energy source, we're either going to be photo or chemo. Okay, so photo uses light, chemo uses chemical compounds, and then this last part is the electron source. This is going to go with along with cellular respiration and how we make ATP, but the electron source falls underneath chemotrophs. Okay, so chemotrophs are either going to be a lithotroph or an organotroph. Litho means we're going to use inorganic molecules, so like hydrogen sulfide. Organo means we're going to use organic molecules to get electrons. Okay, we need to be able to use this table. So let's do an example problem. So what I want you to do is I want you to read the example problem, and I want you to use this chart to figure out how this organism eats. Okay, so the example problem says cyanobacteria use light to fix CO2 into sugars They then break down these sugars for energy in building blocks. Using the chart, how can we classify them? Okay, so we're going to go through this chart and you should have one from each section. Okay, so pause the video for a second, analyze the problem, use the table, and see if you can't come up with the answer. Okay, so let's talk about this. Okay, so the answer for this is that they are autotroph. So they're autotrophic. They are phototrophic. Sometimes we can combine these so that we can say that they're an autophototroph. Okay, or a photoautotroph. I think it goes both ways. Okay, so they're autotrophic, they're phototrophic, and they're organotrophic. So let's look at this problem and see how we got that. Okay, so for cyanobacteria, they fix CO2. This is important. Okay, they use light. Okay, so if they fix CO2, that means they are an autotroph. That means they make their own food. Okay, if they use light, light always goes with photo, so they're a phototroph. Okay, and then they break down these sugars. Sugars are organic compounds. So they break down these sugars, which means that they're organotrophs because they break down organic sugars to get their electrons to donate. Okay, so this is how we'd approach these problems. You should be able to do this on the exam if given the table. Okay, so we have talked about how we obtain carbon, how we obtain energy. Well, we need to talk about the last thing is how we obtain nitrogen. Nitrogen is very integral for living organisms to grow. Okay, our cells need it to make both proteins and nucleic acids. Okay, so if you are a farmer, you're pretty up to date with what you need to do for nitrogen. Okay, so we have a global nitrogen cycle, just like we have a global carbon cycle. Okay, so the global nitrogen cycle is the cycle in which we take nitrogen where it's unusable and we convert it to various forms to make it useful. Okay, so we have a lot of nitrogen in our atmosphere. So nitrogen gas, N2 gas, is what we call inorganic nitrogen. We can't use it. We cannot directly use it for biosynthesis. And it makes up almost 80% of our atmosphere. Okay, so that's a lot of nitrogen that we can't use. So somehow we need to be able to convert that into usable forms of nitrogen. Okay, so some bacteria perform what we call nitrogen fixation. So they're going to take this N2 gas and they're going to convert it to ammonium ions, so NH4+, so which is a form of ammonia, which then can be used by living organisms for biosynthesis. So they can use it to make proteins and nucleic acids. Okay, so we then have bacteria that can take this ammonium and this ammonia and convert it to nitrate, which is we can also use to make proteins and nucleic acids. And then we have other bacteria that can then take it from nitrate and go back to the N2 gas. Okay, so we have some terms that I would like you to know that go along with this cycle. Okay, nitrogen fixation. This is when we go from N2 to ammonia. Okay, so we fix it so we can use it. That's how I remember it. Nitrogen gas gets fixed into something we can use. So nitrification is when we go from this ammonia to nitrate, and denitrification is when we go from nitrate to N2 gas again. Okay, so if you are a farmer, you are very familiar with nitrogen. Okay, so a lot of farmers rotate their crops. Okay, if you're a farmer and you want your crops to do better, maybe they're kind of suffering a little bit, you know that you want to add more nitrogen to the soil in order to get your plants growing better. Okay, we need nitrogen in our soil. In fact, a lot of farmers in our area rotate crops. So one year they'll grow potatoes, next year they'll grow grain, next year they'll grow hay or something. Don't ask me, I just think about potatoes and grain. Okay, and the reason why we rotate crops is so that we can reestablish the nitrogen the nitrogen content of the soil so things can grow better. Now, we can't directly break down nitrogen. So depend on other organisms, specifically bacteria, to do this for us. Okay, so for a farmer, we're going to plant some type of legume, like peas, that have a symbiotic relationship with bacteria that can break nitrogen gas down from the atmosphere and make it more usable. So rhizobia, okay, so these little guys right here, are going to invade the root of pea plants, and they're going to form this nodule. Okay, this is what it looks like on the root. Okay, so when they form this nodule, they go into this symbiotic partnership with the plant. Okay, so the plant's going to provide a place for the rhizobia to live, and the rhizobia is going to fix nitrogen in order for the plant to use it. So rhizobia is going to take that nitrogen gas from the atmosphere, and it's going to convert it to ammonium. And then the plant then can use that ammonia, to make proteins and nucleic acids. Okay, so here we have the nitrogen cycle. So we have nitrogen fixers. This is the term for the bacteria that take nitrogen gas and convert it to ammonia. Okay, we have nitrifiers that are going to take the ammonia and convert it to nitrate. And then we have the denitrifiers that go from nitrate to nitrogen gas. So I would know this. Okay, know this cycle, know that nitrogen fixers go from N2 to ammonia, nitrifiers go from ammonia to nitrate, and denitrifiers go from nitrate to nitrogen gas. Okay, so that's it for 6.3. We have talked about the essential nutrients for microorganisms. We talked about environmental niches. We've talked about the different forms of trophy. Okay, autotrophs, heterotrophs, chemotrophs. Okay, phototrophs. Okay, we now know what this means. We'll be able to use that table. Okay, we talked about the nitrogen cycle. We talked about the carbon cycle. So that's it for 6.3. We'll see you back for 6.4.